Hai Pak Adi, testing.
Halo, apa nih?
Adi dengar ke?
Enggak, dengar.
Oke, clear. Dengar. So nanti Pak Adi akan share how.
Halo.
Halo.
Dengar Cindy kan?
Cindy.
Oke. Oke, sorry. Sound saya itu cukup kuat apa? Saya takut saya perlu gunakan
Boleh
mic.
dengar.
Dengar ya? Kuat kan?
Boleh dengar.
Oke. Kalau thank you.
Halo Pak Adi.
Ya, saya. Yes.
Nanti Pak Adi share screen sendiri untuk share your slide
Yeah. Sure. Yes, I am. I will.
Ya. Buka kamera, banyak orang nak jump testing.
Yeah, sure. Halo.
Saya kena mute.
Habis. Nunggu lama. Gambarnya? Sound putus-putus. Mute. Yo, Aisyah still mute. Masih semua tak. Kenapa saya sendiri yang tak mute? Apa kabar semua? Wah, ini berapa hari hidup di hotel nih, senang sangat. Naik. Hai. Dar. Yes, you. What's up? Oh, gitu. Baik. Oke. Tapi kan saya orang boleh tengok saya nih. Malu, Bo. Oke, baik. Malu. Halo, Prof. Rasman. Prof. Zainura. Dr. Asmi, Prof. Asmi. Tengok senang. Prof. Halo. Wah. Bang Faiz. Mai. Wah, Ini gerombolan si berat ada di belakang semua ini. Oh, itu lebih berat lagi. I hope the connection is good.
Hi, Professor Bear. Can you listen me?
Yeah, I can hear you.
Okay, great. Later you will be sharing your screen.
Oh, yeah, when? 3:10, right?
Yeah, the event will start shortly, and we will pass the screen to you. Hold on for a
Okay.
moment. Yeah. Okay, perhaps you can share your screen right now, and then just leave it at the very first page of your presentation slide.
Okay.
Great, thank you.
Okay. So can you see my screen?
Yes, we can see it. So the event will start, MC and our moderator will start shortly. Thank you so
Okay.
much.
Thank you.
Assalamualaikum and very good afternoon, everyone. Welcome to the day 3 and also our last day of Newton-Ungku Omar Water Symposium and Webinar 2020. For today's program, there will be 3 sessions, which are first session, Challenge in Wastewater Treatment Plants, High Energy Consumption in MBR and Toxic Effects of Emerging Contaminants by Assistant Professor Bae Sung Woo from National University of Singapore. Second session, Domestic Wastewater in Indonesia, the Desludging of Communal Treatment System by Dr. Adi Yuniarto from Institute Teknologi 10 November, Indonesia. Both of these sessions will be streaming on Facebook and YouTube live. And lastly, for the third session, Dr. Sharvini Sivaraman from University Teknologi Malaysia will present the topic Findings on Environment Impact Assessment Study of Decentralized Wastewater Treatment. Without further ado, I would like to invite Professor Dr. Zainurul Zainul Nur to moderate the session. Welcome, Prof.
Thank you, Madam Chairperson. Welcome everyone here, physical participants and virtual audience. Before I pass this mic, so-called mic to Dr. Bae, let me introduce him, his background. So Dr. Bae Sung Woo, I hope I pronounce your names correctly.
Yeah, right.
Graduated from Korea University with Bachelor and Master in Engineering in 2000 and 2002 respectively. He then received his PhD majoring in environmental engineering and minoring in microbiology from the University of California, Davis in 2010. So prior to joining NUS, he was a postdoctoral researcher in the Department of Civil, Architectural, and Environmental Engineering at the University of Texas at Austin. Dr. Bay's research has primarily focused on molecular biology in environmental engineering, and during his graduate studies, he has mostly his work are focused on microbial source tracking for identifying sources of fecal contamination in water. And during his postdoctorate research, the focus of his research was to understand biological mechanisms and functions using new molecular techniques in natural and engineered systems. Currently, Dr. Bay's core research interest is in environmental molecular biology, focusing on natural and engineered water systems, elucidating biological mechanisms, developing biomimic systems, and protecting human health. And by coupling with molecular techniques and environmental engineering to derive sustainable energy saving system in environment, he studies biological systems such as less energy required MBR, eutrophication in drinking water distribution system, ecotoxicology study for emerging contaminants, and antimicrobial resistance genes in wastewater. In addition, his research focuses on rapid detection of microbial contamination in water and development for novel molecular techniques in engineering applications. So his presentation today will mostly evolve around his expertise, and with the presentation entitled Challenges in Wastewater Treatment Plants, Higher Energy Consumption in MBRs, and Toxic Effects of Emerging Contaminants, I pass the mic to you, Dr. Bay. Thank you.
Okay, thank you so much. Kind introductions. I'm very honored to present today's topic, so which is the challenges we are facing in wastewater treatment plant system. So I'm going to give a 2 case study which is we conducted in Singapore. The first case study, which is a membrane bioreactor. So generally, membrane bioreactor require high energy demands. So we are alleviate energy demand using a reciprocation MBR systems. The second case study we conducted through the biological process, some of the emerging contaminants degraded, but some of them did not. How those emerging contamination would affect a aquaculture environment. So the contents of today's presentations. So I'm going to briefly introduce the challenges that we are facing in the domestic wastewater treatment system, particularly 2 main areas, energy and emerging contaminations. So I'm going to give 2 case studies. The first case study, reciprocation membrane bioreactor, which is we demonstrate in a wastewater treatment systems for 10 months period. Another case study, we conducted emerging contaminations, triclosan and methyltriclosan. The chemicals are a personal care product, but also is the adverse effect in aquaculture animal. So we are using the zebrafish embryo as a model organism to study how those chemicals would affect their metabolite and gene expression. So finally, I'm going to make a conclusion and some suggestion for achieving sustainability in wastewater treatment technology. The challenges that we are facing in a wastewater treatment system, particularly domestic wastewater treatment system, which is high energy demand system. For example, conventional activate sludge system, as an example, 60% of energy is for aerations. And also the membrane bioreactor, particularly, they have more than 60% energy used for aerations for not only aeration tank, but also the air scouring to prevent membrane fouling. Typically, the energy requirements, conventional activate sludge a process is required up to 1.89 kilowatt per hour a cubic square meter. A membrane bioreactor need more energy demand. So those higher energy demand, particularly aerations, inactivate sludge and membrane biological process, which is cause a more than up to 40% operation cost. So such a high energy demand, which is leading to a greenhouse gases emissions due to a operation of this high intensive a process. Another challenges we are facing in current wastewater treatment plant, which is emerging contaminants. So those emerging contaminants such as personal care products and antibiotics. So unused or used antibiotics or personal care products, which is a pass through a toilet to the wastewater treatment system. And also agricultural activities contribute antibiotics or personal care products. Those untreated or treated, sometimes those degraded products, which is a huge concern in our society. So for example, this is antimicrobial resistant genes or those personal care products which cause hormone changes in receiving water. Those receiving water often use drinking water treatment plants. Eventually, a human being consume those water. So there is emerging contamination when once it's entered the environment, either a disposal of unused or excretions. So some of the emerging pollutants may not completely degrade at a wastewater treatment system. So today I'm gonna give two case study which is conducted in the US. The first case study, which is a reciprocation MBR system. So MBR, membrane bioreactor, is one of the popular treatment systems for industrial as well as municipal wastewater treatment system, because this system has a small footprint, control the solid, and also provide high effluent quality and good retentions of pathogenic microorganisms. However, a membrane bioreactor has a bottleneck, which is inevitable biofouling on a surface of membrane due to inorganic or organic, sometimes in most cases biological pollutants attached in the surface. In a practical point of view, most membrane bioreactor is operated by air scouring, so using the aerations of aerations, which is prevent mitigate biofouling in the membrane bioreactor. However, those aeration which is cause huge operation cost. Recently, our lab has demonstrated reciprocation membrane bioreactor, which is promising in terms of energy consumptions. So we demonstrate both in lab and pilot scale. So what is the reciprocation MBR system? This is a pilot scale membrane bioreactor, which is demonstrate in domestic wastewater treatment system in Singapore. The short HRT 1.5 hour with chemical backwashing. So simply, this is a reciprocation MBR system. Is the MBR modules are moving forward and backwards. So this is the use of the mechanical imposed shear, so which is allowed to shaking the membrane. So this inertial forces on a hollow fiber membrane modules are prevent biofouling, but also remove some of the nutrients. So I'm going to show you the video. This is the pilot scale membrane bioreactor, which is a reciprocation. So simply a slow frequency of the membranes are moving forwards and backwards. This is allowed to inertial forces to remove the biofouling matter on our membranes. So this figure shows the performance of the membrane bioreactor. So we operate more than 10 months. but I'm going to show you only a 6 months, 108 days. So we actually operations a more than 120 days. We experience a stable performance. Sometimes the manageable TMP increase. This is a TMP. And also this system has a semi-weekly maintenance screening. So we achieved more than 99% COD removal, and we achieved 60% around ammonia removal. And particularly, I want to highlight a specific energy demand for membrane bioreactor, which is those a reciprocation MBR require a very small amount of energies, 0.003 to 0.015 kilowatt per hour cubic meter square at 25 LMS. So which is a 80% of reduced energy consumption compared to a conventional air scalding MBR system. However, you can see there is a linear relationship between a specific energy demand versus a MLSS concentration, because this is a higher MLSS concentration may require more energy demanded. And this plot shows a shift of the microbial community and succession of the biofilms. So several subsequent steps are generally considered to be involved in biofouling process. For example, initial attachments and the formations of microcolony and breaching, succession, and eventually development a mature biofilms. Those mature biofilm which is causing the biofouling event, eventually the human intervention is required to remove those biofouling. In this study, we investigate 6 months and 3 months and 6 months how biofilm could develop on a surface of reciprocation MBR system. One of the example is the microorganisms here, the Enterobacterialis, which is known to produce a signal molecule, which is AHLS. Those microorganisms are increasing during the operation period, which indicate is succession and development of the biofouling, causing the biofouling by the microorganisms. And the difference, the microbial community on a membrane surface we observed. And also we found the succession and selection of the certain microorganisms are causing for biofouling and reciprocation MBR system. We noted that there is the system which is we are using mechanical shear to prevent, mitigate a biofouling, but also we use the chemical cleanings, sodium hypochlorite, which is generally used for industry purpose. So those microorganisms are present after a 6 months period, which means those microorganisms are resistant to mechanical shear and as well as the chemical cleaning. So I'm gonna shift mic here. So I'm gonna share a toxicology study, which is we conducted in our lab. So let's start the background information first. The triclosans, which was chosen as research target compound because this TCS triclosans are antibacterial agents widely used in home and personal care products. And there's external use, a huge amount to use, and also large amount of a TCS was directly discarded into the water system. This TCS is non-volatile and moderated soluble, so that environmental catchment sources include the wastewater, effluent, hospital, and with sludge sample. Even recreation site was the found TCS compound. In terms of occurrence and toxicity of TCS and methyltriclosans, so this is chemical structure of triclosans And generally, triclosan is degraded through a biological, the wastewater treatment system. The byproduct is methyltriclosans. In terms of toxicity, which is known to TCS-induced embryonic lessons and delay of development, or cardiac toxicity, endocrine disruptor. And also the MTCS, methyltriclosan, is interaction with proteins. Distraction is a thyroid hormones actions. And also the phenotype effect has been reported on zebrafish embryo. The occurrence of TCS in the water matrix is up to 2000 in nanogram per liter, and MTCS is up to 300 nanogram per liter. And sediments, even biotas, so has a higher concentrations of a triclosan and methyltriclosan concentrations. So we are using a TCS and MTCS as a model chemical to understand those emerging contaminants affect aquatic environments. So in terms of a methodology, we use a omics approaches. We use a transcriptomic analysis of the gene expressions of a embryo, zebrafish embryos, and we also use a metabolic activities, so metabolites such as sugar, nucleotide, amino acid, and lipid. So using a transcriptomic and metabolomic analysis, we understand how the chemical affect the development of zebrafish embryo. The benefit advantage of the multiomics approaches, which is allow us to better understand the change of genomic and metabolic pathway, and we can predict the biological response to external stressor. In here is TCS, MTCS, and that is increased robustness of toxicity study. And also the zebrafish is a good model organism to predict the human health effect as well. So let's move to a result section first. So MTCS, methyltriclosan, affect a biological pathway of the amino acid synthesis and starch and sources metabolism. This is PCA plot, shows a different concentration compared to control here. And higher concentrations, 400 microgram of MTCS, which is in here as distinct other groups. And also we observe a morphological changes of the pigmentation rate of zebrafish embryo at a 400 microgram liter of MTCS we observe. And also we have the similar a separation MTCS exposure. Then also the heat map shows a 21 different metabolite changes. Particularly this concentration is controlled to lower to higher MTCS exposure. Particularly a higher concentration shows a 21 metabolite, dysregulate metabolite observed. So we also understand a toxic effect TCS, MTCS mixture on zebrafish embryo, because through a biological wastewater treatment systems, the environmental ratio of triclosan versus MT-triclosan, which is 10:1 ratio. So for example, we often found a 10 microgram of TCS, which is indicate a nanogram of a MTCS. So we want to know a real environmental effect of the wastewater discharge into aquatic environment. So as we choose a mixtures, different mixture 1 and 2, 3, meet a environmental the ratio of 10 TCS versus 1 MTCS. The PCA plot on your left shows a differentiate the mixture 3, which is a higher concentration of a TCS and MTCS. And as well as this, the heat map shows a 4 metabolites, particularly a mannose and galactose, glucose, and lysines are interrupted with exposure of a mixture of a TCS, MTCS. So in toxicity of TCS, MTCS zebrafish embryo in a real environment was dominated by TCS mainly because the higher bioavailability of a TCS chemical. And we want to understand the differences in TCS, MTCS, the mixture of TCS, MTCS, and zebrafish embryo. So using multivariate analysis, heat analysis, and as well as pathway analysis, we found a energy metabolism, amino acid metabolism, kill function was significantly influenced by a TCS and MTCS. And also the biological pathway, particularly TCA cycle and nitrogen metabolisms are affected. So those listed a dysregulated a metabolite shows how TCS, MTCS, if they did not treat it properly, gonna be affect a aquaculture animals. Not only a metabolite, gene expression study shows a potential toxicity of TCS. We use transcriptomic analysis and KEGG analysis by the transcriptomics of gene expressions. Particularly, I want to highlight the action of TCS. So oxidative phosphorylations. So this is inner a mitochondrial membranes, which is a TCS act as a proton ionophore. For example, this is a TCS, which is interact with a hydrogens, which is prevent a ATP synthesis. Another effect, the cardiac muscle contraction, which is a similar effect, which is a TCS prevents a calcium a pathway in a cell metabolisms. So this is based on transcriptomic analysis shows how the TCS is interfered at molecular level. And furthermore, so we're using the comparative toxicogenomic database, how a environmental chemical, for example, TCS, affect human health. So using a differential gene expressions shows a potential health effect if TCS exposed to human, what diseases may caused by a TCS exposure. One of the example is cancer, so which is endocrine disruptor or fatty acid synthesis inhibitor. Those a mode of action which is cause a cancer. And also the TCS might cause digestive system diseases because TCS will disrupt intestinal microbiomes in the humans. And also this is a dysregulated phenylalanine, which is cause a nervous system in a human being. So let's move on a conclusions of 2 case study. So the first case study, which is using the reciprocation MBR system, how reduce a energy cost in real wastewater treatment system. So reciprocation MBR, which is generate a mechanical force to control the biofouling. The specific energy demand, particularly this reciprocation MBR, which is ranged from 0.003 to 0.0515 kilowatt per hour cubic meter, which is reduced energy consumption up to 85% when we compare air scalding MBR. One, we noted MLSS concentration, one of the key parameters, control energy consumption because MLSS concentration will correlate a specific energy demands. In case study, a metabolite level was altered at the MTCS exposures, even though when phenotypic change were not observed. In higher concentration is a higher concentration of TCS, MTCS, which is toxicology studies employed. Basically, phenotype changes at lower concentration, environmentally relevant concentration. We could not find any phenotype change. However, metabolite reveal a certain metabolite change due to TCS, MTCS. And particularly, TCS will be dominated effects of environmentally ratio based on TCS, MTCS mixtures. And using the integrated approaches and metabolomics and transcriptomics, which is reveal the toxic effect and eventually potential diseases associated with the TCS and MTCS. Finally, the recommendation or future study for the achieving sustainability in wastewater treatment technology. So we prove the energy saving system in a membrane bioreactor, but still some of the challenges that remain. For example, nutrient removal, nitrogen, phosphorus, and biofouling control, even in resource recovery, such as a fertilizer, nitrogens, and biogas. So thus, we need developed innovative antifouling membrane and also effective or low energy membrane cleaning process are still needed for a domestic wastewater treatment system. For a toxic effect of emerging contaminations, we need environmental risk assessment, particularly for those emerging contaminants in wastewater treatment systems. And toxic reductions of wastewater and also correlation with the treatment process should be considered for design and management and operation of the wastewater treatment system. And that's all for my presentation. And thank you so much for attending and hearing my presentations.
Very much, Dr. B. I forgot to mention earlier that we actually have some time allocated for the question and answer session.
Yep.
So is any question from the online audience or the physical audience here? We are able to perhaps scatter 1 or 2 questions within the next 5 minutes. Anybody? Maybe David, are you there? Would you like to anything?
Oh, there's one question.
Okay, so right.
So how is the transcriptomic data the validate? So we using the transcriptomic data sets, or we have a QC QA control. So transcriptomic data is if they are low quality, we remove, and the high quality the genetic or DNA sequence only used for transcriptomic analysis. So more detail I cannot explain here, but you can go to the paper. So there it shows how to control a transcriptomic data set.
Right. I hope that question, that answer fulfill the question asked by Dr. Yanti. Okay, any further questions from the audience? If no, I would like to ask a question myself. As far as the reciprocal MBR goes, have you actually, I see that it's basically a pilot scale, right?
This is, we have both the pilot and lab scale.
Have both pilot and lab scale. Have you actually tested the pilot scale with actual wastewater, perhaps wastewater from the domestic wastewater?
Yeah, the pilot scale, which is we are exactly same as the same wastewater, which is the wastewater, domestic wastewater in here. There is air squalling MBR. So we are actually have the same operation parameter, which is 1.5, and 3Q return or 3Q intern, 2Q return. So exactly same condition was conducted to compare their energy demand.
So the way the treatment is all in one. Do you require any pre-treatment, or do you use it as a
Well,
polishing
technically,
treatment?
the source of the wastewater is after a activate sludge process. So in here, the Singapore, there is activate sludge process. And the next step is a membrane bioreactor. So without any settling tank, the membrane bioreactor is simply separation of a particle versus a liquid.
Okay. And we have another final question from Dr. Radin Maya. She asked online. So her question, maybe you can read there. What would you suggest to remove xenobiotic organic compound in wastewater? Sorry.
So I think in terms of xenobiotic compounds, I mean, if the water can be used for drinking purpose or any recreation purpose, we might consider to remove a xenobiotic compound, because the receiving water experience a huge impact of a wastewater discharge, particularly those hormone disruptor and the changing the female or male sexes in the fish, and also those are affect a human being as well. So right now there is no regulation, but hopefully we have the regulation to remove further xenobiotic compounds, particularly those antibiotics or toxic compounds in domestic water.
Right. I think that's all the time. Thank you, Dr. Bay. I think that's all the time that we have for the first session today. So if the audience are here, or the virtual audience have further questions, perhaps you can get in touch directly with Dr. Bay. Maybe you can leave your email there in the chat section so that people can connect.
So I can type my email.
Yeah, you can type your email there. And we thank you again, and we will send you the e-certificate as a token of appreciation. Originally, for your information, we would like to have our speakers here giving the presentation in person, but due to COVID-19, so everything has to be done online. But thanks again, Dr. Bay. It's a pleasure.
Thank you so much.
So you may stay for the next presentation, or you may leave, or whichever they prefer. We would be very happy if you would like to stay.
Okay, thank you.
Thank you. Okay, it gives me great pleasure to introduce our next speaker, Dr. Adi Yuniarto. So Dr. Adi Yuniarto is actually currently working at Institute Teknologi 10 Surabaya, Indonesia. He obtained his Bachelor of Engineering from the Department of Environmental Engineering of the same institute where he's currently working, and also his master degree. He obtained his PhD, however, at UTM from the Faculty of Chemical Engineering in Environmental Engineering. He is currently lecturer at the Department of Environmental Engineering. He used to hold the position of the chairman or the head department of the Department of Environmental Engineering in ITS. So his research focus is in wastewater treatment, pollution control, environmental engineering. And he has also, actually his research study also more or less evolved around the same topic as Dr. Bay earlier, which is in MBR. So his presentation today is entitled Domestic Wastewater in Indonesia, the Desludging of Communal Treatment Systems. So without further ado, Dr. Adi, over to you. Thank you.
Okay, thank you. Thank you, Professor Nura. Good afternoon, everyone. And thank you for introducing me to the forum. And in this wonderful session, I would like to give you some overview about domestic wastewater in Indonesia, especially about the desludging system happen commonly in Indonesia, and one particular example in one region in Indonesia, Gresik. Okay, I'm sorry. Is this okay? Hopefully, my talk will take about 20 minutes. Yeah, hopefully, yeah. Now let's see the main point of my presentation today. First is decentralization domestic wastewater in Indonesia, and the second point is about septage treatment system, and the last one is the desludging practice of communal sewage treatment happen in Gresik Regency. I want to give some overview about Indonesia so you can feel how difficult the challenge, yeah, for sanitation and wastewater system in Indonesia. The population of Indonesia is about 273 million right now, and 56% live in urban area, expecting 67% live in urban area in 2030. We have 8 major islands, but 58% population live in Java. It's quite small. Here, we have decentralization governments with 34 provinces, 415 regencies, and 93 cities, which this local government can set up the local regulation, but for sanitation and domestic wastewater infrastructures funding, these governments still depend on national programs and national agencies. Let's see the existing sanitation situation in Indonesia right now. Wastewater, especially the grey water from a house or home or from the public facilities, still disposed directly into the water bodies, especially to the drainage system. The latest report say that more than 75% of house already have individual toilets, but most of them in low quality latrine. Open defecation still happen. In some part of Indonesia, shower and washing in stream still exists. In a rural area, in an urban area, illegal disposal, dumping of sludge or septage from desludging of septic tanks happen to the to the river or to the empty space, especially in a rural area, and so on. The impact of poor sanitation, we already know that 70% or more rivers and groundwater are polluted. The sad one, 50 to 1,000 per 1,000 babies mortality due to diarrhea, which is 4% of diarrhea happen to the population. 30% children under 5 years old are stunted, and we have to pay 25% higher price of pipe drinking water. The losses for this situation, poor sanitation, potentially about $4.2 billion per year. And to overcome this situation, actually in 2007, the national government already set up the programs for sanitation, and we try to have 100% sanitation access in 2030. Mainly the program done by ministry. The first program is community-based total sanitation, is belong to Ministry of Health. The second one, the infrastructure development, belong to the Ministry of Public Work and Housing. And the acceleration of sanitation development program from Ministry of National Planning. We have also program from Ministry of Village and Transmigration. We call it Village Funds, and special funds and grant for sanitation, Ministry of Finance. This is also including the sanitation fund from program from donors parties. For example, USA, JICA, Danida, yeah, and foreign parties. And the last one is sanitation of school, because from Ministry of Education, because we have also have to improve our school condition. Target in 2019 is we have 85% of improved access sanitation, and 15% of basic access, and 0% of defecation. However, the progress is happen like this. The last situation is we only achieved 77.4%. I'm sorry for this decimal. About 77.4% for improved access of sanitation, 15% of basic access, and no access was 7.5. But if you see the progress, yeah, in the right graphic, we see there's some good progress, especially for these last 2 years. And from the data, we know that the urban access is quite no improved. Yeah, but in rural, we have a good improvement during these 2 years. Yeah, and we have also the national program for community-based total sanitation, or we call it STPM, starting in 2008, and consists of 5 program: ODF, open defecation free, and washing hand with soaps, safeguarding drinking water and household food, and domestic waste management, including the solid waste, and domestic wastewater management. And the program, especially for ODF, for sanitation, the last reports show the good progress. Yeah, we still have about 7.6% people still do the ODF. Okay, let's talk about the domestic waste management. The concept is depend on based on the regulation of Ministry of Public Work and Housing. The new one is number 4 in 2017. We have to system, the on-site system, which is a septic tank and the communal treatment, up to 2 to 10 house connection, and a shared toilet. This is practice here, and we have desludging trucks, and they will move the septage to the septage treatment facility, usually about 15 to 20 kilometers away from the city center. And the second one is offsite system. We have a house connection and a sewerage system, and go to the full offsite treatment. We have 3 kinds according to the regulation. 3 kinds of sewage treatment plant. The full scale or city-wide STP, more than 4,000 house connection, or 20,000 PE. And a small scale STP around 10 to 4,000 house connection, and a specific area STP. right now in 2019, we have on-site treatment about 216 septic treatment plan. Because the end of the treatment is septic treatment plan. We have 260 plus. I don't know the exactly. Some data say 262, 264, but around 260s already built and rehab optimized until now. The offsite treatment in regional scale or city scale, we have some cities already connected. Some of them is not all, the whole area of the city, maybe some most part of the city, Medan, Parapat, Batam, Tangerang, Jakarta, Denpasar, and we will have 5 more big system for sewerage system and STP in Jakarta, Palembang, Pekanbaru, Jambi, and Makassar. Jakarta will about 2 million PE. So offsite treatment using decentralization and the communal system, we have more than 50,000 community-based communal system, and for institutional base is around 240, built in 128 city or district. But the capacity is not so big, around 1.7 million population. PE, 1.7 million PE. So it's about, if compare based on data in 2017, only around less than 2% of population have offsite system. Let's move to the next part, septic treatment system. Okay, according to the regulation, it is mandatory to individual septic tanks or institutional or communal system to be desludging. Some local government through public work agency, already developed the system. So the customer have to apply using Android application to set up the time and to apply and put some schedule in, and it is mandatory to desludging once every 2 or 3 years, and they will transfer the sludge or septic to the septic treatment plant, usually 15 to 20 kilometers away from the populated area, because the smell and people try to put away. And decent capacity of septic treatment plant is about, the small one is 15 cubic meters per day, and the biggest one is 300 cubic meters per day. This is the system. This is the system that recommended by the regulation. The desludging system start from the inlet ditch and go to the sludge separation chamber, SSG. This is one of intellectual innovation of Prof. Johnny Hermana, and actually this is the work is like a filter, so the the solid keep on the surface and dry it up naturally, and transfer to the sludge drying bed and become the fertilizer and cover soil. And the under drain for the low capacity sewage treatment plant, usually they use anaerobic bio reactor or anaerobic buffalo reactors, and go to the facultative maturation pond and apply to the wetland before the goes to water bodies. The high capacity one, they use extended aeration sometimes oxidation ditch, sometimes aeration tank, extended aeration tank. They have go to clarifier and wetland before to water bodies. Overview of FST, actually the figures is like this, yeah, commonly like this. So is the trucks, emptying the tanks and go to the filter. The function is separating solids and sludge, liquid through filtration and decanting processes. And the advantage is simple, no skill operator. But the disadvantage is we have to change the filter, usually from gravel and sand to maintain the efficiency of the filtration and prevent clogging, and transfer the sludge from SSC to sludge drying bed is manually or using crane, and requires a large area, especially for bigger sewage septic treatment plant. They have to build more chamber for SST and naturally drying process. So usually they need about 5 to 12 days. And so the design principles is the solid separation chamber generally used and sand and gravel. Yeah. The thickness about 20 to 30 centimeters, and there is a kit for remove the supernatant, and we have to remove also the sludge, drying sludge. So you can see this video later. You can find in YouTube. This happened in Gresik. So they have to apply or in trucks will come and go to the sewage treatment plant. They empty the tanks. Yeah. After coarse screen, the liquid comes to SSC. After 5 to 12 days, they will take the sludge, dry sludge, go to the treatment to fertilizer, and the under drain water comes to the ponds and goes to the river. Here's some operation and maintenance happen also in Gresik. After after emptying the tanks and the liquid comes to come into SSC and process drying, they have to manually remove from the SSC to STPs. Yes, and they have dry or clean SSC before new sludge come. Okay, this is some practice of septic treatment management in Gresik. Regency of Gresik, located the north part of Java Island in east part, have 1.3 million in population, and the contour or the area is hilly, limestone clay soil. The average altitude about 25 meters from sea level, and this is an industrial and fishery area actually, and housing with mostly individual septic tank. They have 134 on-site communal STPs with one septic treatment plant, which is Pitoyokuchi. This is the location of STP Pitoyokuchi. Here, north part of populated area, about 25 to 20 kilometers far away. That 134 communal STPs located in 10 districts, and 9 coming in 2020. And they mostly use anaerobic buffer reactor system with 10 or 6 to 10 compartments, most built in kampungs in the city center or surrounding district. Yeah, you know, the very dense area. One STP usually for 15 to 60 house connection, built by local government, but funded by Ministry of Public Work and Housing. All of communal sewage treatment plant operated by local company. Right now, the condition is moderate. Some of them very good condition. Effluent is not regularly monitored with the local government. Effluent is turbid due to high level of sludge. And we found that 12 STP located in narrow street or alley. Yeah, you see the situation is quite good because it's 4 meters wide of street, but on the next one is quite narrow. Yeah, this one is only 1.5 meter wide. So this process happened in Gresik. Okay, we clustering the location of STPs. We can use trucks if the road is about more than 4 meters. Yeah, usually the trucks capacity, so they can handle 3 times per trips. We were sent to the septic treatment plant. The cluster 2, only for 2 to 4 meter wide of alley or street. We use 3 wheel motorcycle already modified for septic tanks. We call and the capacity is about 1.5 to 1.8 cubic meters per motorcycle, and transfer to the trucks, which is standby at the nearest location. But for cluster 3, which is I think is three communal septic tanks, communal sewage treatment. We use kedot tank. Kedot tank is stand for kereta dorong septic tank or septic tank push car. Yeah, operate by men. They can push or they can connect with motorcycle. But after near the location, they have to push it, and they will come back. The kedot tank, this push cart, capacity is about 1.1 or 1.2 cubic meters. Sometimes if full of sludge, they cannot use all capacity because too heavy. And then they go to the trucks. Sometimes in one day, in one location, they have to go back 2 or 3 times, and do the sludging. Okay. To summarize what I have to say so far, the progress sanitation program in Indonesia is very good. I'm not say it is excellent, but is almost ideal for us, and still as the top priority. And the funding is not affected by the crisis right now, financial crisis or pandemic, and involving community to care about environment and sanitation still happen. Yeah, local government make some program that involve the community, like right now is about clean city, health kampung, and so on. And the last one is empowering local government and community to maintain and operate the existing facility, because the local government, most of them have a financial budget, but most of final budget and in salary. So for maintain and operate is sometimes they not put it on the top priority. Yeah, that's all. I think I fully give you more information about the decentralization wastewater in Indonesia. Thank you very much for this opportunity.
Thank you very much, Dr. Adiniarto. We have a few last minutes for questions, but we do have two questions from the online participants. One first question from Professor Graham. So yes, you can either read the question yourself. You can read that. You want me to read it for you? And we have another also questions from Professor Graham. So yeah.
Okay. Yes.
So I guess the question is how actually, yeah.
Yes.
Want to go ahead and answer
thank
those
you.
questions?
Okay, I'm sorry. Okay, thank you, Prof. David Graham. For monitoring the community sewage treatment plant, we still use, method that very conventional. Then they have to take the samples and bring to the local agency, and the local agency will analyze in a local appointed laboratory, and they have to report per month actually. But unfortunately, they still ask the local government to local community to pay, because the facility already give to the community. The community asks money, take some fees from the host, from the connection, and also for the analyze fee. And we still ask for the local government to make it free, but they still happen with that we have to pay. The local community have to pay.
Yeah, if I could respond, that's very helpful. We were doing something in Nigeria, and we actually created a reward program, or they did. We were just advising, but the group in Nigeria and Lagos created a reward program for local communities based on how well their system was working. So if the way it worked was if their system wasn't working well, they had to pay. And if their system was working well, they didn't. And it was quite a nice inducement for people to try and make sure their system, and there was some community pride that developed. It's just a thought, but the problem you describe is a really big problem. That you've got the systems are the right systems for the purpose, but obviously it's a disinducement because people have to pay to monitor them.
Now we educate the local community just to see the effluent by physically. If there is turbid, they have to report to the local agency, especially the environmental local agency, and ask for desludging. Desludging is free. Desludging is free. But for testing the quality of the effluent, they still have to pay. Actually, it's not expensive. I think only around $40 US, $40 US for some parameters, but they still have to pay. And now so local government gives some excuse. They have to report around about 6 months, every 6 months, not every month. But yeah, hopefully.
That's very interesting. Thank you.
I'm sorry, Prof. The connection is not so good.
One last question.
Okay. Dr. Raj,
yeah.
okay.
She was asking.
Radin? Oh yeah, yes. Okay, Dr. Radin, laundry. Yes. Yeah, laundry discharge, especially from washing facility, become attention. Yeah, lately in Indonesia, especially in urban area, yeah, around ITS maybe, because they in small store, a small house, but produce extensive laundry wastewater. So since this year, let me explain sample Surabaya. Surabaya, the local government of Surabaya already come out from some regulation that every laundry station have to have pre-treatment and using septic tanks or anaerobic buffer reactors. But for small store, they have to prepare some rapid sand filter. So you're a simple one, rapid sand filter. We still conducting some research about the achievement of this regulation. Yeah.
All right, thank you.
Thank you.
I think that's all the time that we have for you today. That's the end of session. The second session for today. Again, I would like to convey my gratitude to Dr. Yuniarto for joining us. And if anyone have further questions, you can directly get in touch with Dr. Yuniarto. Thanks again. We will send you the e-certificates. So the next session will be a closed session discussing the finding, the outcomes of the Newton project. So those in the Zoom session, you may stay, but then we have to say goodbye to audience joining through FB Live and YouTube. So thank you again for joining us. We'll see you next time we have another program.